All right, so last class we learned about the process of transcription translation, and we started with information in the DNA, which we will make a copy of in the form of pre-mRNA. This is premature mRNA, and this is called transcription, which happens in three steps. We have initiation, which is the binding of the RNA polymerase to the promoter. And we have elongation, which involves using the template strand to build the complementary pre-messenger RNA. And we have termination. Okay, so once again, in this process, you're going to have a coding strand, which, for example, if it's ATTCCG CAT, then the template strand is going to be complementary to this, which would be TAA GGC GTA, just following base pairing rule. And when you build the messenger RNA, the pre-mRNA is going to be using the template strand to create the complementary strand. Okay, and of course, if you do this, then the coding and the pre-mRNA is going to be identical, but thymine will be replaced by uracil. Okay, so this happens in the nucleus, and it's the first step in creating the protein. It happens in the nucleus. From the pre-mRNA, we are going to um, modify it and change it into the actual mRNA. This is called post-transcriptional modifications. Which involves three things: RNA splicing, okay, removing the introns and connecting the exons. Number two, we have addition of 5' cap. And number three, poly A tail on 3' end. Okay. Then this also happens in the nucleus. Then it is going to go into the cytoplasm where we will convert it into protein. And this is translation. Okay, and this is where you're gonna take the mRNA and you're gonna convert the codes into amino acids using the codon chart, right? That I gave you last time. This is the codon chart. Okay. So let's just as a warm up, I guess. Right, I'll take the pre-mRNA here. AUU, CCU, CAU. Just as a warm up, can you please convert it into amino acid? Okay, go ahead, do that. Just let me know when you have it. Just type it out. Oh, you guys don't have the codon chart, right? Is that the problem? You didn't print one out? Uh, that's probably it. Here, I'll put it on the screen for you. You're gonna need one for today. Okay, you're gonna need one for today. There we go. Okay, just look it up. AUU, anybody? Do you know what AUU is?
MET? MET?
AUU is not MET, but good try. Let's try it one more
Oh,
time.
sorry. This is ILE.
That's right. Yeah, so AU, which is right here in this box. AUU is ILE. And then the next one is CCG.
Pro.
CCG is right here, Pro. And finally, we have CAU
His.
is his. Yeah, so this is very short. It doesn't begin with MET, it doesn't end with stop, because could be like a middle of a section or something like that, right? But that's translation. Okay, so I'm gonna go through the process of translation with you. Um, I mean, it's up to you if you want to draw this or not. Actually, you know what? This next part you can draw, and then after that, it's kind of hard to draw because I will be moving things around. But this next part you can draw. Okay. And this is the structure of tRNA. Okay. Now, for the process of translation, which is going from mRNA to the protein, there are three kinds of RNA that we need. Okay, and they're all listed in the PowerPoint slides. I'll go through them with you quickly, and then I'll explain more. The messenger RNA, the mRNA, is the copy of the gene that contains information about the sequence of amino acid. Basically, the mRNA is the recipe. Okay, this is the recipe that we're going to be using. Recipe that we'll be using to build our protein. Okay, the rRNA, which stands for ribosomal RNA, it's part of the ribosome, right? Long time ago in lecture 3, we learned that the ribosomes are needed to create the peptide bond between the amino acids and therefore build our polypeptide. And the ribosome itself is made up of rRNA as well as some protein. So you can think of the rRNA as the chef, okay? That's actually going to be cooking the recipe, so to speak. But we need the ingredients, okay? And the ingredients are going to be brought to the ribosome by transfer RNA or tRNA. Okay? And you can think of this as like the helper that brings the ingredient to the chef, and the chef will cook the meal according to the recipe. Okay, so I'm gonna draw the transfer RNA with you to show you what it looks like, and we're gonna go from there. First of all, you should know RNA is single-stranded. So this is like a single-strand RNA. It has a 3' end, and then it has a 5' end. Okay, and what happens is this actually folds up, and it will look something like this when it's folded up. Right? Maybe I'll just do it over here. Folds up, and then it will look like this. So I have a 3' end here, and then it has a 5' end here. And it's the same strand, but it just loops back on itself. And it's able to stay in this shape because on the within the strand, there might be some complementary bases right here. So I'm going to zoom in a little bit for you. And I'm just going to make up some letters here. Let's say this is A, C, U, U. There's no thymine. We're talking about RNA here. And then later on in the strand, maybe this is UGAA, and they're able to form hydrogen bonds to stabilize the structure. Similar over here, you might have A, AACC, and then UUGG here. And again, you can form hydrogen bonds. Same thing over here. Okay, so these are complementary nucleotides from different parts of the RNA, of the tRNA. And it just happens that they are complementary. And so you're able to pair them up and stabilize this 3D clover leaf structure. Okay. Now at the 3' end, this is where it will be carrying an amino acid. Amino acid. Remember, the tRNA is responsible for bringing the correct amino acid to the ribosome so that you can build the protein, right? And there, that is the 3' end is where you will be transporting the amino acid with. Okay, I'm gonna give you a moment to catch up, and then the next part I'll try to explain. Okay, let me do a little bit of rough work here while you copy that down, and I'll explain afterwards. All right. So let's say we're gonna have the mRNA now. Okay, so this is the mRNA. We already cut out the introns, and we have the poly A tail and the 5' cap and so on and so forth. All right. Now for simplicity, I'm just gonna have a few nucleotides here. You know what? Let's draw it straight. Okay, so it's easier to follow along, I hope. Right. So I'm gonna have a few letters here. You can copy them down. It's no big deal. AUG. Okay. There's supposed to be no space here, but I'm gonna put some space just to make it easier for us to see. GAU, totally making these up. And then on the last one is like that. Okay. Now I'm gonna draw some transfer RNA here. Okay, this is the part where you might not want to draw necessarily, just because it is going to be a little bit messy. I'll post the notes, of course, so you can decide what to do with it afterwards. Okay, but give me a moment. I am going to have to copy this guy. Okay, let's see if it's gonna work. There we go. Okay, so this is gonna be the transfer RNA, and at the bottom of the transfer RNA, we can add it over here. There is a triplet of nucleotides that are particularly important. So let's say this is AAA. Okay, so this is called the anti-codon. Okay, I'll explain in a second. So let's say this guy has the anticodon AAA here. Okay, I gotta write it better. AAA. Right? Just give me a second. I gotta set this thing up. It's really hard to do in a virtual environment, but I'm trying my best here. So bear with me. There we go. Let's bring this guy back in. Okay. So we have the AAA here, and then I'm gonna put another guy right here. This one is gonna have, and you can copy these down if you want, okay? CUA, and I'm going to put another one here. And then this is going to be UAC, UAC, and then just one more here. This is going to be CCA. So each of these transfer RNA is going to have an amino acid on them. Okay. And I'm going to tell you what the amino acids are in a second. Okay. But this is the messenger RNA. These are the tRNA. And we're going to have the ribosome here. You might remember the ribosome has two subunits. There's a large subunit at the top, and then there's a bottom subunit, right? It's like a hamburger, like clams onto the mRNA. And there are actually three slots here in the ribosome. You might see that in some of the drawings online, but we don't really have to worry about it. All right. This is the part where you should just watch and not copy yet, okay? So over here, the first codon is AUG. If you look up AUG over here, I'll put it over here so you can see it. If you look up AUG, it's the start codon, and you should memorize the start codon is MET, right? So these things are going to try to bring the amino acid over. And you just kind of have to believe me for the moment that this one is going to be MET, and this one going to be MET, and then this one is going to be PHE. Okay, we'll figure out the other two later. So let's say this one is going to try to come over here. Okay, this tRNA is going to bring the amino acid over here, and it is not showing up because this guy is in the way. Let me see if I can put this move to the back, order, send to the back. There we go. Send all the way to the back. Can I do that? Order, send to the back. There we go. Yeah. So the tRNA is going to come over, and it's try to put it over here, right? But as you can see, this is the wrong tRNA, right? AUG, we don't need the amino acid PHG, we need the amino acid MET. So this is the wrong one. But how does it know it's the wrong one? Well, it's the wrong one because the codon that's on the mRNA is not matching, it's not complementary to the anticodon of the tRNA that's coming in. And if they are not complementary, then this thing is wrong, and then it wouldn't fit. It's like the wrong key. So it's going to flow away. Okay? So let's try another key here. Let's try another tRNA. So this guy is carrying the MET, and then the anticodon is UAC. It's going to come over here, and it's going to dock. And is it working? This one works, right? This one is complementary. Right? UAC is complementary to AUG. So if the anticodon and the codon matches, then this is the right amino acid, and it's going to stay. Okay. Najia, just hold on. Okay. After I explain, you will understand. Okay. So over here, let's see the next one. Next one is UUU. UUU is the codon, right? So can you guys look up UUU on the codon chart here? You don't have to tell me. You just look it up and see what it is. See for yourself. Look up UUU over here. Okay.
PHE.
Yeah, sure. Right. Thank you. UUU is PHE, right? So let's just, for the sake of discussion, this one's gonna try to come over here, and is it gonna fit? Nope, it's not gonna fit because UUU is not complementary to CUA. The anticodon does not pair up with the codon, so this is just gonna bounce off. Okay, but what about this one? Is this gonna fit? Well, this one is. Oh, I took along some of the other stuff as well. Just give me a moment. Okay, so I'm gonna take this guy, and this one's gonna come, and now it's gonna sit here, and it's gonna fit, right? AAA is complementary to UUU. And if it fits, then what happens is the MET will get transfer to the PHG. And this one has done its job, and it's now going to move away. It's going to, let me try to do that again. This guy is done, and it's going to move away. It's going to move away. It doesn't let me move away, but there we go. It's going to move away along with the anticodon. Okay, anticodon is going to go with it. Okay. So now it's empty. It will have to reload. It will have to pick up another MET, right? Okay. And so let's see. Next one is GAU. Which of these transfer RNA is the correct one to come along? Is it the one on the left or is it the one on the right?
Right.
The one on the this one? This one is correct, yeah. This one's gonna come, it's gonna bring the amino acid and then it's gonna sit right there, right? Okay, but what is the amino acid here? What's the amino acid? So this is where it gets a little bit tricky, okay? Please listen carefully so you don't get it wrong on the test. When we look up the amino acid, that's carried by the tRNA, we do not look up the anticodon, okay? We do not look up the anticodon. Instead, we have to look up the codon. So if you see the anticodon as CUA, when you look at it on the table to figure out what amino acid it's carrying, we're not going to look up CUA, okay? This is called the codon chart. So you have to look up the complementary codon. What's complementary to CUA? Complementary to CUA is GAU. Okay, so we're going to look up GAU. GAU is right here, which is ASP. Okay, so if it's the right one, then again, watch, please. The growing one will break off and then it will get attached to the new one, not the other way around. Okay, the growing chain breaks off and it gets passed on to the new one. Then this one is done. This tRNA is done and it's gonna basically leave and it's gonna go look for another amino acid. Okay, it's gonna look for another PHE in this case. Do you guys understand? I mean, this is not very good animation, I suppose. If you want animation, you can look at the videos I post. But I hope it conveys the information to you. Are you with me? Give me some, feedback, questions, or whatever you have. Thank you. Now, then the ribosome, and this is where it's really hard to draw, right? The ribosome will actually move forward. It'll move forward, and then it will read the next set of codes, right? Did you have a question, Khadija?
Yeah. You told about that we don't need to see the anticodon.
Yeah, we do not look up the anticodon if we want to
Anticodon.
figure out the amino acid.
We see the codon first, and then what is happening in the cell. They see the codon, and right amino acid is come. Then the anticodon. We don't need to think about anticodon.
Well, okay, so like on the test, right? For example, let me see if I have a question here. Okay, I do. 1 second. Okay, so hold on. I gotta put the table in for you to see. You will have the table as a reference on the test, okay? So don't worry about memorizing it or printing your own. There we go. So let's say the tRNA has an anticodon of CUU. Okay, I'm gonna do the polls here for you guys. Okay, what amino acid is it going to be carrying? Please try that out. Let's change one of them. There you go. What is the answer? Uh-oh, I only have 4 answers. Where's the rest of you? Do you guys not understand? If you don't understand, you gotta ask, okay? It's not something you will figure out yourself.
Is it leucine?
Well, let's find out, right? I'm waiting for people to choose their stuff. Okay, so if I draw the tRNA, I mean you don't have to draw on the test, but I'm drawing it for you. I'm trying to figure out the amino acid, and then it says the anticodon is CUU. Okay? You do not look up CUU, okay? Because CUU is the anticodon. Right? We do not look up the anticodon. Right? This is called the codon chart. So you have to think about this. Like if this is on the mRNA, if this is mRNA, what is this going to bind to? What's the codon that will correspond to CUU? Well, corresponding to CUU is going to be GAA, right? GAA. So this is the codon that we need to look up. GAA. That's GLU. So the correct answer is E.
How do you know GA? I don't understand.
You don't understand why you look up GAA and not GUU? Is that the question?
Yeah,
Okay. Do you understand the difference between codon and anticodon?
yeah.
Okay, codon is the code on the mRNA, right?
Yes.
And the anticodon is the code on the tRNA. What determines the amino acid that you need to have to build? Is it the codon or is it the anticodon?
Codon.
That's right. That's why we do not look up the anticodon. We have to look up the codon. How do you get the codon from the anticodon?
Yeah.
The anticodon is complementary to the codon.
Oh, okay. I
Yeah.
got it.
Okay. That's why CUU is GAA. All right. So back to our notes here. Right here, we have a tRNA with an anticodon of CCA. What is the correct codon? What should I look up to figure out the amino acid? Okay, everybody should look it up and then see what the amino acid is. Thank you, Melissa. Anyone else? What amino acid is this thing carrying? So for CCA, again, that's the anticodon. The codon would be GGU. You use the base pairing rule. GGU is right here, and it's GLY. Okay, so what's gonna happen to this thing? It's gonna flow over here. It's gonna sit right there, and it fits because it's complementary. Then the growing strand will break off, and you will form a new peptide bond. And then this thing is gonna flow away. I'm trying. This thing's gonna flow away, and it needs to be reloaded. Okay? And then we'll move on to the next one. What is this one here? What's UAG? Anybody remember UAG? What's
Stop codon.
that? That's stop codon. So stop codon has no tRNA corresponding to it. Okay, so when it reaches the stop codon, no tRNA will come. Then this means it is the end. And then this amino acid will get this protein will get released, and then the ribosome separates, and then there we finish building the protein. Okay, so this is the translation
That means
process.
in this way, the amino acids are sequenced.
Sorry, one more time. Yeah,
Amino
yeah, this is the amino acid sequence. That's right.
sequence. Yeah.
Mhm. Okay, so I'm going to show you the picture here, which hopefully will kind of make sense. Okay, so first of all, the tRNA, some of them are empty because they've just delivered their cargo, right? So then they have to load up with the correct amino acid. Okay, so this one here, the anticodon is GAG, which means the codon would be CUC. And you will see that LEU correspond to CUC. So it's going to pick up the LEU. It's not going to pick up the SCR. Same thing over here, the AGU, which corresponds to UCA, and then it will pick up the SCR. So each transfer RNA will only pick up a specific amino acid. Okay, you're not gonna have this guy picking up PHE or something. Okay, it doesn't work, right? Okay. The identity of the amino acid has to be complementary to the anticodon. And of course, an enzyme will help attach the amino acid to the transfer RNA. Then this process has already been happening for a while in this picture, but you can see the ribosome has a large subunit and a small subunit, and it has been moving along the messenger RNA here. Okay, 5 prime to 3 prime. And what happens is the amino acid chain is growing. This one has just deposited the latest amino acid, so it's on the way out. Okay, on the way out, and it will need to pick up another amino acid. Right, there are a lot of amino acid floating around in the cytoplasm. It's gonna get loaded up with a new one, and we have an incoming transfer RNA with the corresponding amino acid. It's going to go here at the front. Then this whole chain will be transferred onto the ARG, right? And then this one will exit, right? So new one comes in on one side, old one exits on the other. And then in the middle, that's where you're going to be growing the new chain. And it will keep on going and going and going until you hit the stop codon. When you get to the stop codon, no corresponding tRNA will be available. Then you release the protein. Okay, each of these green things is an amino acid. Right? The protein can be long, can be short. Some proteins are hundreds of amino acid long, and the ribosome will separate, and it will find another mRNA to build the protein from, and that is translation. Um, Seriously, any questions before we move on? Okay, I hope not. So what is this? Is this lecture 7? This is lecture 7. I think I put the notes in the wrong place. But it doesn't matter. Here is the um study guide, and I think there's a question here. I'm just gonna do one of them with you guys to make sure you understand what's going on. Okay. So here it says complete the table below. I'm gonna move the colon chart over here as well. Um, it's not really letting me do it. Let me try one more time. There we go. Oh, there it is. Okay. I know it's a little bit small. Hopefully you have your own copy, then you're not gonna have this problem. There we go. Okay. So here we have the coding strand, and we're gonna write the mRNA from the coding. Okay. How do we do it? Anyone want to help me out here?
Copy
Just copy.
without T. T will be U.
Yeah, just copy it, right? Charmaine, you're correct as well. Right? Just copy it. Right? Coding strand and mRNA are the same. So we're gonna go A, but because we are doing RNA, you have to change the T to U. So AUG, I just copy everything. Read the instruction carefully on the test and the quiz. If it says key it in with no space, then key it in with no space. Remember, you cannot use your notes this time. So please study and remember the stuff. Okay, next. Yeah, the codon chart will be available as a reference sheet, like your chemistry class, okay? It wouldn't be like multiple tabs. We only have one reference sheet on for biology, right? So it would just be one extra tab with the codon chart right there. So this is the mRNA. What about the anti-codon? Well, if you take a look at here, the codon, which is on the mRNA, is complementary to the anti-codon. And the anti-codon is on the tRNA. If it helps, you can write down tRNA here, right? You probably need to have some blank paper with you on the test so you can, work it out. It's hard to see it on the screen and do it at the same time, right? So bring some blank paper. Although, as I said, if you're writing things out, make sure you tilt your camera so that we can see what you're writing on, okay? And take pictures of it at the end. Over here, this would be UAC, UAU, GCU, UUA, CUA, AUU. Okay, remember the relationship between mRNA, between the codon and the anticodon is that they're complementary. Complementary. All right, here comes the amino acid. When we look up the amino acid, remember to look up the codon and not the anticodon. So we're going to look up AUG. Okay, look up AUG, which is MET. AUA is ILE, so on and so forth. Okay. CGA, AAT, AAU rather. AAU is ASN. GAU. Okay, remember, we're looking this thing up. And UAA is stop. Okay. Please write the word stop if it's a stop codon. Don't leave it blank. If you leave it blank, they will not like we're not going to be able to give you mark for a blank. Okay, you have to put the word stop in it, and that is it. Do you want me to do another example? Just say yes if you do. Okay, if not, I'm going to move on. All right. So there are more practice in the study guide. You can do it. Amino acid corresponds to the coding strand. Yes, it does, but don't look up the coding strand because the coding strand has T in it. This thing has no T, right? So always look up the mRNA. The mRNA is where the codon is going to be. The mRNA is the codon. Right? So look up the AUG. Don't look up ATG. And look up AUG. Any other question? Okay, there are two things that's due this week. Quiz is going to be on Wednesday. It's all of lecture 6 and 7. We'll finish 7 today. And there's also the microscopy lab, which will be due on Friday. Okay, it's going to be due on Friday. I'll talk about the lab on Wednesday, I guess. All right. I will continue. The next thing we will learn is called mutation. Okay, mutation. And I think I'm just gonna do it over here with the note. It's easier for you to understand, I'm hoping. Okay, what are mutation? Mutation is permanent changes or change in the DNA. Remember, DNA contains the instruction to make the protein, and if you change the DNA, you're gonna end up having possibly a wrong protein. So, mutation. There are two kinds of mutation we will look at. And we will look at base substitution. And then we will look at insertion or deletion. Base substitution is when a single nucleotide is changed. And then there are three outcomes. Silent, missense. A silent is also called neutral, okay? Just write it down first, I'll explain. Neutral. Missense or nonsense. Insertion is addition or removal of nucleotides. Okay, could be one, could be two, could be three. You're actually deleting things or adding things in it. And then there are two results. Multiples of three or not multiples of three. So like if you add 3, 6, 9, 12, that's called multiples of 3. If you're adding 2 or deleting 1, deleting 5, adding 7, these are not multiples of 3. It is generally worse if it's not multiple of 3. It can lead to something called frame shift. I will explain. And then over here, no frame shift, but you have extra or missing amino acid. Just write it down. I will explain. Hey, give me a sign when you're done copying, okay? Like I can never tell. Okay, just give me a thumbs up or type me an okay, done or something. Like in class, I can see people.
The third number of base substitution.
What's that?
The third number of base substitution.
The third number of base substitution? Like
Yes.
here? What does it say?
Yeah.
It says nonsense.
Nonsense.
Nonsense. Okay. I will explain what they mean. Okay, let me know when you guys are done. All right. Just gonna wait for a few more dones and then we're gonna go. You can do it. You can tell me you're done. All right. Ideally, you have this thing with you, like now. Okay? If you don't, I guess you can just look at mine. I'll make it bigger. Now, the mutations can be caused by various reasons. Okay, it could be just mistake that you made during DNA replication. Certain things will increase the risks of mutation, like exposure to UV light, certain chemicals exposure will increase the likelihood of mutation, right? And the mutation has to be in the DNA for it to be permanent. If we change the mRNA, you're just going to have a bad batch of proteins. But remember, mRNA is not permanent, right? mRNA will get broken down. So if you have a change in mRNA, that's okay. The problem will be transient. Okay, once you make a new mRNA, it should be fine. Having said that, it is difficult for students to understand if I make the changes at the DNA level and then track it to the protein level. Okay, a lot of people have trouble following that. So I am gonna make the change at the RNA level for you to understand first. Okay, but know that in reality, the changes has to occur in the DNA. If the DNA is wrong, the RNA is also going to be wrong, right? So let's say we have here, it's called the wild type or WT. This wild type is the normal one. Okay, it's the before you have the mutation. Okay? Wild type, the normal one. So for the sake of its discussion, let's say the normal one is UUU. On the DNA, it would have been like TTT. Again, I'm doing it at the RNA level so that you can understand better. So let's say the UUU, what would it be in a protein? Please look up UUU. Hey, don't wait for me. You gotta look up. For this to make sense, you gotta look it up yourself.
Phenylalanine.
Yeah, it is PHE. Right here, right? Right here. So we're gonna have the mutant. The mutant is the one with the mutation. That's what it's called. So we're doing base substitution, which means you are only changing one nucleotide, and I am going to change this last one here. I'm going to change UUU to UUC. Okay, some mutation occurs, there's a mistake, it changed the last nucleotide to a C. What is the what is the new amino acid? Please look it up. What's the new amino acid?
It is also
The
PHE.
same.
It is also phenylalanine.
Yeah, it is also PHE, right? UUU becomes UUC, same amino acid. Okay. If you're gonna have a mutation, you want to have this kind of mutation. Why? Why is this mutation not too bothersome?
As the amino
Because
acid
amino acid same.
Yeah, it's the same amino acid, right? It's not gonna affect you, right? Okay. So no change in the amino acid, no change in the protein sequence. You wouldn't even know about this, right? That's why this is called a silent mutation. Okay, this is number one, silent. Okay, or neutral. You wouldn't even know about it. Okay. I forgot if I asked you guys this question before, okay? Have I asked you what percent of your DNA is same as mine? Forgot if I asked you that. No, maybe? Okay, I guess not. Okay. So what percent of your DNA is same as mine? Okay? As far as I know, we're not related. Okay? So your DNA, if you take a look at all your ACTGs and then you compare to all my ACTGs, what percent of the DNA would be the same? How many? Sorry? Anyone? I've only heard a few of you speak before. Okay, maybe someone who hasn't said anything. Give me a number, right? Your DNA, my DNA. What percent would be the same? 10%, 20%, 30%?
Nothing.
Nothing? 0%. Okay. Anyone else? Anyone else have a different idea? What percent? No? No more? Let me pick on. Yeah, go ahead.
I think more than 90%.
More than 90%? Okay, yeah. So we have 0%, we have more than 90%. Anyone else?
Because just of, I think 1 or 2% are
Just
different.
1 or 2% difference. So 98%, 99%.
Yeah.
Yeah. Yeah. Good, good. Yeah. You and I, in fact, you and anyone else on this planet is 99.9% identical. Okay. It might be more identical if you're talking about siblings and relatives, but you and I, okay, even though we're not related, but because we are both humans, we are 99.9% identical. The differences between you and I are actually caused by these silent mutation. Okay. So you might have a UUU, I might have a UUC, right? But they code for the same amino acid, and yet our DNA is different. Okay. So these little single nucleotide differences, it's called SNP, SNP. SNP stands for single nucleotide polymorphism. We will talk more about the significance of SNPs in what is it? Lecture 9. Okay. My other class, someone asked me, okay, we are 99.9. Do you know what percent of us is same as bananas? Okay. Between us and bananas, the fruit bananas. Right. If you look it up, it's actually 60%. Right. So we we are more alike than we think with other stuff. Anyways, let's do missense. Missense. Okay, so again, wild type, this time UUU, once again is PHE. And we are going to change, this time I'm going to change the middle one. Okay, it doesn't matter which one you change. I'm going to change the middle one. I'm going to change it to UAU. And what is UAU? UAU, yeah, it's right here, right? Tyrosine, TYR. So that's a different amino acid, right? That's a different amino acid. It's a mistake. That's why it's called missense mutation. Okay, missense mutation. Now we're going to talk about whether this is bad or not bad in a bit. But let's take a look at the third one first. Third one is nonsense. Like all this stuff we are learning is nonsense. Okay, we're going to go UAC and I'm going to mutate it to UAG right here. Can change the last one. So let's look up UAC. UAC is right here. It's TYR, tyrosine. And what is UAG? What is UAG?
Stop.
That is a stop codon. Right? Stop codon. Right? So that's nonsense. Now, which one is worse? Right? Is missense worse or is nonsense worse? Well, the answer is it depends. Depends on several things. Depends on number one, where it happens. Okay, so the effects of let's say the severity of the mutation depends on number one, location. Okay, type Right? So let me explain. Let's say I have an amino protein, okay? Protein right here. And it's supposed to be, this many amino acid long. Okay. So then, MET, TYR, blah, 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 whatever, okay? And then it's gonna stop right here. Let's say we have a nonsense mutation here. Nonsense. Well, what does it mean? Well, if I have a nonsense mutation here, it means I'll make the first three and then I will stop because nonsense creates a stop codon, right? Is that bad?
Yeah,
Yeah,
because the beginning it will be stopped.
yeah, exactly, right? It's pretty bad because at the beginning it stops and I will not be making the rest of it, right? So this protein is not going to work, right? But what if you have a nonsense here? Right? Then it's not as bad. It could still be bad, but not as bad because you have a chunk of the protein made already. You're just missing 2 amino acids, right? Okay, so this is worse, and then this may be okay. I'm not saying it's going to be fine, 100%, but it's not as bad as this one. So location matters. Okay. Another thing about location is, let's say this is an enzyme. You remember enzyme, right? We talk a great deal about it. Right? This is an active site. Active site. If I have a missense mutation here, missense, versus a missense mutation here, right? Then which one is worse? Is missense in A, missense here worse, or missense B worse?
A.
A is worse because A is in the active site. If you change the amino acid to something else, it might change the shape of the active site. This is worst. And then this may be okay. Maybe okay. All right. It's all like where it's happening. Think about like a car. If I say there's a nail in your car, and like there's a huge nail this big stuck in your car, is it bad? But like is it gonna be detrimental to your car? Well, it depends on where the nail is. If it's stuck in your tires, then yeah, that's probably not a good spot, right? Your car is not going to function for too long. You're going to get a flat tire, right? But if this thing is like stuck to your trunk, I mean, it still damages your car, but it's not going to affect the functionality of the car. You can still drive it around with no problem, right? So mutation is like that. It depends on where it happens. Okay. In some places, it's going to severely impact the function. Other places, not so much, even though it's the same kind of mutation. Right? Even though it's the same nail, depends where you poke it, it's going to have different outcome. Do you understand?
Yes.
Yeah, okay. Also, when we change, this was a long time ago. Let me see if I still have the slide there, right? Look. There are different kinds of amino acids, right? We learned some are polar, some are non-polar. So let's say we have a missense mutation, right? If you replace a non-polar one with another non-polar one, then maybe it's fine. They are both non-polar. They have similar properties, right? It's not going to be a big deal. But if the mutation changes a non-polar one into a polar one or vice versa, then that could be a big deal, like the hemoglobin, right? The mutation changes it from a polar one to a non-polar one, and then you end up having sickle cell anemia, right? So the type of mutation matters. So in general, you cannot say missense is worse than nonsense and nonsense is worse than missense. It really depends on the context. Okay, that's that for now. Moving on. So that's base substitution. Silent is the best kind to have. Missense, nonsense, not so much. Now we're going to talk about insertion and deletion. But before so, let's go back to the PowerPoint slides for a second. This is everything I was telling you about. We wrote this in the notes already. Here are some more examples you can go through yourself. Sickle cell anemia, right? Polar one to non-polar one. It changes the shape of your red cell. We talked about this before. Okay. Now we're going to talk about indels, okay, or insertion deletions. When you add or remove nucleotides that are not multiples of 3, it can change the reading frame. What is a reading frame? Well, the reading frame for ribosome is in triplets because the codons are made up of 3 nucleotides. So when the ribosome comes, it can only read 3 things at a time. So like the sentence, right? The big cat saw the fat dog. Right? It's all 3 letters. I understand the sentence perfectly fine. There's no, mistake or whatever. However, if we delete the B here, then the sentence becomes the, which is fine. And then the next 3 letters would be IGC. And then the next 3 letters would be ATS. And you see, like everything after the point of deletion makes no sense anymore because we shift the reading frame. This is called frame shift. Okay, so if you delete an amino acid, if you delete a nucleotide, all the codons beyond that point is going to be wrong. It's going to be different codes. And if the codes are different, the amino acid will also be different. But what if I delete 3 letters? We take out the whole thing right here. Okay. I can still read the sentence. The cat saw the fat dog. Okay. The sentence structure is maintained, but I lost some information. Okay. I no longer know the cat. Like, was it the big cat? Was it the fat cat? Was it the red cat? I don't know. Okay. But I still have the sentence structure maintained. Right? So if you delete 3 nucleotides, reading frame will be intact, no frame shift, but you might end up losing an amino acid. Now, is that bad? Again, it depends. If you lose an amino acid in the middle of an active site, that could be bad. If you lose an amino acid at the beginning of the strand or end of the strand, maybe it's okay. Depends what kind of protein we're talking about. Over here, the difference is you added an extra letter. If you add an extra letter, again, you change the reading frame. Everything after the point of insertion is different. Right? Over here, we're adding an extra 3 letters. Now we have an extra word, but again, the sentence structure is maintained. So that's what I mean by the reading frame. Okay, if you maintain the same reading frame, then it's usually less problematic. If you do not maintain the reading frame, then you're going to have what's called frame shift mutation. So let's work through this sheet here. I'm sorry, I'm going to skip the break today. Lots of stuff to do. Just want to make sure that we cover it all. This is question 11 from the study guide. Okay, if you haven't been printing out the study guide, you really should for lecture 8 and 9. Okay, lots of things from lecture 8 and 9 that we need from the study guide to do. So here it is. I guess I can put it here. I don't know if you can see it. Maybe move order center back. There we go. Okay, so this is the original DNA. It is the template, I believe. It says so in the question. So here, I'll write template. Okay, so take a moment and write the mRNA sequence and the amino acid sequence. You can chop it up to make it easier to follow along. I'll let you do that, please. If you have the amino acid sequence, please send it to me. Okay, I'm going to give you a few moments to work on this. Okay, so that's not the difficult part. I'm hoping you have it. I'm gonna write down for you. Okay, make sure you do complementary base pair here to figure out the mRNA sequence. If you don't understand, you need to ask now. Otherwise, the rest of the class will not make any sense to you. I know I keep on saying that, but it's true. Okay, and then we're going to go ahead and find the amino acid sequence. Does anybody have the amino acid sequence here? Okay, thank you. A couple of people giving me the answers. Matt, TRP, ASN, ARG, CYS. Stop. Everybody got that? Okay, so next we're gonna have a mutation here. It's the same sequence, but with a change. We can chop it up into triplets, and you have to compare the letters one by one. Can you find the mistake? Where is the mutation? I'm gonna pick on someone here on the list.
Second codon,
Oh, sure.
ACC, ATC.
Which one? I'm sorry.
In second codon,
Yeah, the second one, right? Second one is it says ACC in the original, but
ATC.
now it's ATC, right? And so we're going to go ahead and do the transcription. TAC will still be AUG. Instead of ACC now is ATC. So the new one is going to be UAG. The rest will be the same. So I'm not going to write it. You can write it later. What will be the amino acid sequence? First one is AUG, so that's MET.
Stop.
And then stop. That's right. Stop. Will there likely be effects? In other words, do you think this is bad?
Yeah.
Yeah, this is very bad. Very bad. Very, very bad. Right? Because you lost the protein. Right? Now listen to me. Remember those talks about red light and green lights from lecture 6? What if this is a red light protein? Right? If it's a red light protein, and then you have a nonsense mutation, then you're not making the red light. That's one hit. Okay? So you still have a backup copy. But if this happens again, then you have 2 hits in the same cell. That could cause uncontrolled cell division. Do you understand? So this creates a stop codon. What kind of mutation is this? This is a nonsense. Nonsense mutation. And it leads to, I forgot to teach you this word, truncated protein. Truncated means shortened. Shortened. Truncated. All right. I'm going to give you a moment and try the next one. 3 minutes maybe. Please do try it. Just message me if you don't understand. Okay, private message me or something. I'll explain again. But you need to know this stuff to do the quiz. All right? Not kidding. All right. So again, you would chop them up. It's easier when you chop them up. Take a look here, please. If you chop them up, you see there is an extra one that wasn't there before. Before it was like, evenly divided into triplets, right? But now I have an extra one, which means somewhere along the way, I've added an extra nucleotide in the mutation. So let's compare it one by one. Don't forget to compare to the original, okay? So TAC, totally fine. And then ACC, but this one is GAC. Okay, so there's the mutation. The mutation is that we have an extra G right here. Okay, it's not a base substitution. It's not like I changed the nucleotide. It's you actually add an extra one. I know I added an extra one because there is a additional one at the end, right? Like it's shipped over. So what's the new amino acid sequence. On the new, actually, you know what? I got to do the new mRNA first. New RNA would be AUG again. And then the next one would be CUG. GAA, CCG, CUG, CUG, and then just A. Amino acid sequence, I'm going to look up AUG, which is MET. After MET is CUG. I'm going to cheat. I have the answer here. MET and then LUU, GLU. You just look them up, right? There's nothing to it. Pro, and then LUU, and then LUU again. Right? And then the A, I mean, you can use dash instead if the line is too confusing. It could be confusing for some people. And then there's a single letter A here, but remember this could go on for some time. So dot 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 dot. Is this bad or not bad? Can you tell whether it's bad or not, or like it depends? Huh? Sorry.
Maybe the last, maybe it's not bad.
Maybe not bad. Okay. Anyone else? Anyone think this is bad?
This is bad for me.
Well, how come it's bad, Shamin?
Protein is different. Maybe the sequence is different. So
Yeah, the
people
sequence is
know that-
different. Very good. Look, see, the original is MET, and then TRP. This one, the mutant, only the MET is the same. Everything else is wrong. Right? We get a completely different protein here. I don't know what this protein is going to do, if it's going to do anything at all. Right? So will there likely be an effect? Yes. Okay. Bad. And what kind of mutation here? It is an insertion. And then the insertion is going to result in frame shift. Right? Because we only insert one. And this frame shift will lead to multiple missense mutations. And not only are we changing the frame, all the amino acids will be wrong after that point. And yes, it's bad. This is a completely different protein. Different sequence. Okay, do we need to do one more or you guys get it? Just let me know. I'll do whatever you want. One more or no? Okay. Well, there are 3 more in the study guide. You can try it. The answers are posted. All right. Make sure you have a good understanding of this. Okay, just a couple more things from the PowerPoint, and then we're gonna finish up the lesson. Give me a second here. Okay, just 4 more questions, in fact, and then we're done. I will put it right here.
In exam for this, we need more time.
Yeah, maybe. Let me see. Yeah, let me see how much time did I give you guys? Course. What are you guys? 201. Quiz number 3. It says 50 minutes for 25 multiple choice question. That's 2 minutes per question. Is that like not enough? 25 questions, and there's no short answer this time. For 15 minutes. What do you guys think? 25 questions, 50 minutes.
Make it
Do you have any multi-select question?
Multi-select? No, not on the quiz, fortunately for you guys. Yeah. What do you guys want? Tell me. If it's reasonable, I'll do it.
Can you make it 60 minutes?
60 minutes? Okay. Everybody happy with 60? Yeah. Anyone else? 60?
Yes.
Sure, I'll make it 60. That's fine. I'll do it now before I forget. 60. I don't really care. It doesn't matter. 60, 50. It's the same. Hold on. 60. Done. Yeah, 60.
Thank you.
Yeah. Good luck. Okay. We are almost there. Almost there. Let's take a look at these questions. It's a different codon chart, but it's the same. Use it the same way, okay? Same way. Create a base substitution in the coding strand. So now this is DNA. This is coding. That will result in a missense mutation. Okay, missense mutation. So this is how you do it. Yeah, you can pick anyone. It doesn't matter. Okay, just pick your favorite one. Okay, let's say we pick this one. Okay, so then let's write down the mRNA. If it's AAC in the AAG in the coding, then the mRNA is AAG as well. It happens to be the same. If you pick something with a T, make sure you change the T to the U, right? Okay, so missense. Now, how do I do a missense mutation? Well, AAG, if I look it up, AAG is LYS. If I want a missense mutation, I need something that's not LYS. And then the easiest way to do is like change the first or second one. So let's say I change the first one to C. So now it's CAG. CAG is going to be GLN. That's a missense mutation. But don't forget to change it back in the original DNA. So that's going to be C. Okay. Like I said, you can pick another one. I'll do another one here for you. Let's say you do GAT. GAT is going to be GAU. GAU is going to be ASP right here. GAU. It's going to be ASP. So give me a missense mutation. Missense mutation. What are you going to change? Are you going to change to G, change to A, or change to U? There's no fixed answer, okay? There are multiple correct answers here. Which one should we change? Yeah, I'm going to change the A. Sure, let's change the A. We can change the A to C. We can change it to other stuff. So GCU. What is GCU? GCU is ALA. Okay, that's a missense mutation. Next, create a base. Sorry. Create a base substitution in the coding strand. Again, this is coding. And we need a nonsense mutation. So mRNA, amino acid. Okay, so what's a nonsense mutation? Nonsense mutation means I need to have a stop codon, right? So stop codons are UAG, UAA, UGA. And we wrote it down last time, right? So these are stop codon, which means on the coding DNA, on the coding strand, it would be TGA, TAG, and TAA. because coding and codons are the same, but U and T are swapped. So I have to look for something here that will allow me to create one of these stop codon. Okay? So over here, for example, this says CAG. So if I change the C to a T, then it's a stop codon. If you are not sure, you can change it to mRNA, and then you will see it stop. Okay. Not every codon can be changed into a stop codon. Okay. Like this one is not going to work. This is AAT. I cannot change a single nucleotide to create a stop codon. Just not possible, right? How about this one? Can I change a single nucleotide and make this into a stop codon? I want to have a stop codon because I want a nonsense mutation. Which letter should I choose to make this a stop codon?
Instead of A, we can put U.
Which A? The first one or second one?
Yeah, first one.
First one, right? So we're going to change it to a T, right? Because remember, this is DNA. And then, if you try, you will see that it's like that. Again, you don't have to do both. You just have to do one of them. Okay, just one of them. All right, next example. Okay, this is template strand now. Template. And we not want nonsense. So let's write down the stop codon again. UGA, UAG, UAA. And this time we want the template. What would the stop codon look like on the template? Stop codon and coding are the same, but stop codon and template would be complementary. So UGA is actually ACT. UAG would be ATC and ATT. So I have to find something here.
Last one.
Which one? I'm sorry?
Last one. ATT.
Okay, yeah, but here's the thing, right? The last one is already a stop codon, right? So it's already going to stop anyways. So if I change it to a stop codon again, like it's not really a mutation in that case, right? So which codon can we modify to create a stop codon? Well, you can do it right here. Instead of AAT, I can change it to ACT. Okay, if I do that, let's take a look at the mRNA. Template to mRNA is complementary, so ACT will become UGA, which of course is a stop codon. Okay, this is amino acid. Now, the amino acid is supposed to be, the protein is supposed to be 1, 2, 3, 4, 5 amino acid long. If you stop early, you are losing 2 amino acids. So that's like 40% of the protein, right? Even though it's towards the end, but 40% is a significant loss. It's probably going to be bad. Okay, last one here. We're going to create an insertion in the coding that will result in a truncated protein. So truncated is nonsense. It's shortened. It's shortened because it stops prematurely. Okay, so once again, the stop codon, UGA, UAG, UAA, and on the coding, it will look the same, but we change the U to T. So over here, I have to insert something, insert a nucleotide that allows me to create a stop codon. So that would be a good place to put it. Right? When the ribosome reads it, it will be like ATG. And instead of GAA next, it's going to read like TGA. And TGA is a stop codon. Okay, you can also try to put a T right here. It's going to be TAA. That's it. One last question from the PowerPoint. I'm just going to copy it over. Which of the following is likely the most deleterious? Deleterious means very bad. Let's see. Deletion of AAA at the beginning of a gene. So this is multiple of 3. So not too bad. You're going to end up losing 1 amino acid. Okay. Lost 1 amino acid. It's all right. I mean, it could be bad, but doesn't look like it's the worst. Okay. Let's check the other ones. The codon UAC is mutated into UAU in the middle of a gene. So let's see UAC and UAU. UAC is tyrosine. UAU is the same amino acid. So what kind of mutation is it? What do we call it if it's the same amino acid?
Silent.
Silent. Yeah, silent mutation. The best kind. Okay, so this likely has no effect. Okay, no effect. Okay, so probably not the first two. UAC is mutated into UAG. We know UAG is a stop codon near the end of the gene. Right? So you will lose couple of amino acid. Again, it might not be the worst. I'm looking for the most deleterious, the most harmful. It's happening nearing the end of the gene. It's going to stop anyways. I'm just stopping a little bit earlier. Maybe it's fine. Deletion of an A, of a A nucleotide at the beginning of a gene. Okay, so what does this mean? This is not a multiple of 3. You're gonna have like frame shift. Okay, multiple missense. Everything's gonna be wrong after the deletion. You're gonna get a whole new, different sequence. That could be pretty bad, right? So I'm betting on D, but let's just to be safe, check the last one. Insertion of UAA at the end of a gene. UAA is a stop codon, and again, it's happening at the end, so probably not as bad as D. Okay, again, I'm not saying the other ones are not bad, but the question is asking you for the worst, okay? And D is definitely the worst out of all these. Okay, that is it. Study hard. Okay, there's another question. In regards to base substitution, we use T instead of U. Well, no, because like the question here, these are all DNA, right? That's why we are using T. Okay, the changes has to be happening in the DNA. So anytime you change the DNA, you have to be using the T. But if you are just working the questions out like this, when it's the mRNA sequence, then you can keep on using U. Does that make sense? Okay, study hard. We do have class on Wednesday, so please attend. I don't know your schedule, okay, but maybe wait until after the class before you do the quiz, right? No need to rush to it. You have until midnight anyways, right? Okay. When you're rushing through things, you tend to do worse. Very difficult lecture for number 8. Okay, please print the notes, print the study guides, and we'll go through it together. And we'll also talk about the lab on Wednesday. Okay. Have a good one. Bye for now.